Good evening, everyone, and thank you for joining us for our third Seeds of Knowledge webinar. We begin this evening by acknowledging the traditional custodians of the lands on which we meet today and pay our respects to their elders past and present. I extend that respect to Aboriginal and Torres Strait Islander peoples who are joining us this evening. We'd like to acknowledge that tonight Laura will be joining us from Bidjigal country, and that the Seedbank that Laura represents as part of Botanic Gardens of Sydney is located on Dawarral country. Apologies if I've mispronounced those. I'm joining from Wadawurrung country, and Seeding Victoria itself is based in Djadja country. I'm Elle. I'm the Partnerships Coordinator here at Seeding Victoria. Tonight marks the halfway point of our Seeds of Knowledge program, with tonight being the third of 6 webinars, which have been very generously funded by Bullock and Northern Grampians Landcare Network. Our 2 previous Seeds of Knowledge webinars can still be accessed via our collaborative YouTube channel with Moorabool Landcare Network. If you search Moorabool Landcare Network on YouTube, you'll find the channel, and please feel free to share. The Seeds of Knowledge program is a truly collaborative effort, helped by organisations who share our goal in making knowledge free and accessible for everybody who's interested. Sharing knowledge is the keystone to project success, and by joining the minds of academics, practitioners, experts, locals, and the community, we can help create a field of people who can access and share skills. I'd like to warmly welcome Laura Watts, the Seedbank Collections Development Coordinator at Botanic Gardens of Sydney. I've personally followed Laura's work for a while, not creepily, I hope, via socials, and I was excited when she agreed to present for us tonight. So thank you, Laura, and I'll pass over to you.
Awesome, thanks so much, Elle. So I am going to be giving you a bit of an overview of the Seedbank and Conservation Collections program that we run at the Botanic Gardens of Sydney based at Mount Annan. My name is Laura Watts. I'm the Collections Development Coordinator there. And basically, to give you an idea of what our program actually focuses on, it's securing seed for the future. So you may be familiar with the Botanic Gardens of Sydney site that's represented by Royal Botanic Gardens of Sydney based at Darling Harbour. So this is the eldest of the 3 sites, but there's another 2, one based in Mount Tomah that is a curated collection of cool climate species, and another that's based out at Mount Annan, which is mostly Australian native species, and that's also where the plant bank where I work is based. So the Royal Botanic Gardens, the Botanic Gardens of Sydney, is one of the oldest scientific institutions in Australia, and it hosts significant scientific expertise. So we have the National Herbarium of New South Wales, the Australian Plant Bank, the Research Centre for Ecosystem Resilience, Living Collections, Plant Clinic, and Education Services. Research Centre for Ecosystem Resilience, if you're unfamiliar with that, that is our team that has a genetic focus, and we've been working really closely in collaboration with them in recent years to really improve the approach that we've taken to seed collecting. We also have been recently benefited by the National Herbarium of New South Wales relocating from the Sydney site to the Mount Annan site, so we've been working closely with them as well. All in all, the idea of the Botanic Gardens of Sydney is that we're basically a one-stop shop for plant conservation of all sorts. Pretty much all aspects of the all components of plant conservation we can cater for in-house, from the living collections focus to, genetics and seed banking, plant ID, plant pathology, etc., to science communication of those efforts. I mentioned the Australian Plant Bank, which is where I work, and unfortunately I can't really give you a behind the scenes tour, but I thought that you might appreciate some of the imagery of what it actually looks like so that you can sort of visualize throughout this presentation. You can see the central top image is actually a view of inside the seed vault. So those little silver packages are actually what we keep our seeds in, and they can stay in those packages for decades to centuries under the right storage conditions. So that's sort of a long-term aim of the conservation collections program. So there's all sorts of steps that have to take place prior to that to secure those collections. We also have an X-ray machine, so you can see to the not the far right, but just in between, there's a little X-ray of some seeds, which gives us some great insight into the quality of the collections that we're making. And we also have, obviously, some really beautiful living collections, tissue culture, which you can see down the bottom. And we also use cryogenic storage as sort of a newer research technique for storing some of the exceptional species that aren't orthodox like some of the others and won't store under usual conditions. We also on the far right have the zigzag aspirator, which is just one of the fun pieces of technology that we have at the Seedbank. It's actually from agriculture initially, but it basically just separates out seed and other material from a collection based off weight by blowing air through that like little zigzag piece that you can see there. So that's just some of the fun things that we get to play around with, and hopefully you can keep that in the back of your mind while I'm talking. So the Australian Plant Bank, the main aim that we have is to conserve germplasm of Australian plant species. So germplasm just refers to any part of the plant that we can use to reproduce, any material that we can use to reproduce that plant in the future. And so we have a focus on New South Wales and Eastern Australian species. We have a seed bank. We also have a tissue culture collection for things that can't be seed banked. We utilize cryogenic storage as a backup for some of our seed banked collection, but also for things that don't bank under orthodox conditions. And we also have laboratories and seed and plant research equipment that's highly specialized. And we use that to do research into germination and dormancy, seed storage biology, conservation of exceptional species, restoration and translocation, and climate resilience. So like I mentioned, my role as the collections development coordinator is actually a focus on the acquisition side of things. So obviously part of having these collections is that someone needs to actually go out and make those collections. So the seed bank team is primarily responsible for that. And like I mentioned, we do have a focus on New South Wales species. When the seed collecting program began in the 80s, there was less strict legislation around this in the different states and territories. And so we do have some collections from other states and territories. However, now most other states and territories have their own botanic gardens and seed banks that are responsible for this instead. But basically, our focus is to collect unrepresented taxa and multi-provenance diversity. We also have a focus on collecting threatened species, but with maternal line collections, which is essentially just a more robust form of seed collecting. And we make plant tissue sampling collections as well. We have a real focus on long-term conservation storage and viability monitoring. So that's because we are a conservation collection seed bank. So we really focus on quality over quantity, which means that we don't sort of focus so much on having, really large seed orcharding and things like that, and producing huge quantities of seed, but rather focusing more on the species that require more in-depth research and just ensuring that we have representative genetic material captured for as many of the New South Wales native species as is possible. So we also look into the distribution of seeds, so living collections, and we collaborate externally outside of the Botanic Gardens of Sydney. And we support in situ management and translocations as well, especially with threatened species officers that are part of our local department offices. We facilitate and take part in national and international seed conservation training, and we're also a duplication facility, which means that we send our seed to other botanic gardens and seed banks to have a backup in case of, the worst case scenario, and we also receive seed for that reason too. So why use seeds at all for ex situ conservation? The reason is because seeds are the plant's natural storage device for genetic material. Many seeds can be stored in a small space, so they're cost effective and they're space efficient. Seed may be stored for long periods of time. This includes acacias, eucalypts. Some of these species can last under the correct storage conditions for centuries. The costs are relatively low and there's little maintenance required. So a large amount of genetic diversity can be captured in one collection as long as you are making those collections appropriately. So I mentioned I'm part of the Seedbank and Conservation Collections team, and that our purpose is to secure genetically representative collections of all New South Wales flora, accompanied by comprehensive scientific data to support conservation efforts and ensure the long-term preservation of our native plant diversity. But how do we actually go about doing that? So we will go out and we'll collect seeds across species distributions. So that means multiple populations, usually the extent of the population for however far it spreads to. We have a collection strategy that's backed by genetic data as much as we can. So that's working in collaboration with the RISA team, which is our genetics team at the Botanic Gardens of Sydney. And we do our best to understand the factors that influence the germination niche of those species. So temperature, moisture, fire, etc. And this means that we not only have these collections, but that they're actually useful. So in our perspective, there's no point in going out and collecting these seeds and removing them from the natural environment if we're not then going to follow up on that and figure out in 10 or 20 or 50 years time, if we need to actually utilize this seed, how we would go about doing that. And a lot of the time, collecting the seed can actually be the easy part of the job, and germinating it is actually quite difficult. So our role is to prevent extinction of species, keep species protected till threats can be managed, provide material to support research in other parts of the gardens, but also externally, and provide material to return species to the wild, which is the ultimate goal. Obviously, the seed bank is a bit of an insurance policy, and it works best if it doesn't exist in isolation from the rest of the world. So we have a very active collection. It's always being accessed by external parties and inside and outside of sort of government and department offices to better the chances of these species surviving in the wild. So a little introduction to the rest of the team, because it's bigger than just me. So we have Nathan Emery, who's the manager of the Seed Bank and Conservation Collections team, and Graham Errington, who's the Seed Bank curator. And then we also have a bunch of great seed bank officers who are on externally funded projects, and that's Kat Thompson, Steph Carusi, Sam Paget, Ruby Perossian, and Alex Thompson, although I just noticed that everyone's names got a little bit skew-iff, so sorry about that, guys. Cool. So there's quite a complex process to actually going through the motions at work and figuring out how we are actually going to conserve these species. There's a lot of species out there, and one of the questions that's really vital to ask at the beginning of all of this is if there's even seeds available. So we have this little conservation framework that our manager worked up for us that has been really helpful in sort of communicating or putting into words for us what it is that we're actually doing, but also communicating to others the complexity of what it is that we do. Because a lot of people think, you go out and you collect seed and that's, job done, pretty easy. But like I've been saying, it is a little bit more complicated than that, because maybe seeds are available, but we don't know the genetics. At that point, maybe it's better to go through and fund a genetic study first. But there's so many different pathways forward, and you have to be really careful about how you proceed, because we know a lot about many of these species. We know much more than we used to, but there's still much that we don't know. And so, yeah, it's a bit of a work in progress. And as you can see, I won't go through the whole framework because it is quite complex, but just to give you an idea that it's more than just kind of going out and grabbing some seed and putting it in a box in the back of a freezer. Like, there is a lot of thought and deliberation and decision making that goes into specifically what it is that we're doing and what we're working on and why. So one of the things that we do as part of our team's work is we'll do a viability assessment. So all of the seeds that come in through the seed bank will do a baseline germination test to check the viability of the seed. And we also do standard and multifactorial tests, like I was mentioning about fire and temperature conditions. So there's a lot of environmental conditions that will trigger germination, and they're all sort of part of the mystery of things that we focus on at work. And we also have a range of seed testing workflows. So like I mentioned, we have the standard sort of viability and dormancy focus, as well as post-fire germination response. And we look at thermal optima as well, which is for maximizing germination and feeds nicely into thermal climate modeling, which can predict germination under future climates. We've also been doing water stress germination studies, which show sensitivity to drought, and we investigate seed storage behavior. So we're really lucky as a team. We get to do a lot of really interesting things. And there's sort of something for the field geeks and something for the lab geeks. Myself, I'm a little bit more on the field side of things, so we'll get into that shortly. So just to give you a little overview of what it is that we actually have stored in the seed bank. So in terms of New South Wales species that we hold, we have around 2,938 of New South Wales species, and that's represented by 8,000 accessions. And so that just gives you an example of the fact that we will actually make multiple collections of the same species in order to collect across the distribution and ensure that we're collecting to maximize the genetic diversity of our seed collections. And we also, because we've had a reasonable amount of funding in this space, we have 72% of our threatened species that are represented by at least one seed collection. So that doesn't necessarily mean that they are what we call representative collections, meaning that they have multi-provenance, multi-population collections. But we do have some seed for the 2/3 of our threatened species in New South Wales, which is a great effort. And that's really because we've had a conservation focus for over 20 years, and in the last 5 years we've really had very specific focus on conserving New South Wales threatened species. So the seed bank switched to a primarily threatened species focus in the mid-2010s that was funded through Stage 1 of Saving Our Species. It was the start of a great partnership which we've continued up until today and into the future. The focus was initially on obtaining 60 species per financial year, and we often exceeded that number. However, we'd run into issues with reproductive material being unavailable, meaning that we'd go out and the seed wouldn't be available for us to collect. The species taxonomy might be uncertain, so occasionally we have made collections of species that turned out after some genetic testing to not actually be distinct species. So that's one of the reasons that we've prioritized genetic testing prior to making seed collections. As well as low fecundity and low viability, which are understandably big issues for threatened species, because there is, of course, a reason why they're threatened, which is mostly that they're pretty bad at reproducing. So sometimes we would only be able to secure as few as 5 or 10 seeds for a particular species. Now, the thing about that is that if you know how to germinate a seed, that's not the end of the world, because you can do, that's 10 plants that you could put out into a translocation site. However, unfortunately, with a lot of these threatened species, not only do they not reproduce well, they also tend to have complex dormancies. So it's a fairly complicated space to work in. And at the end of the day, what we got to was that we didn't have enough seed to be able to consider most of our collections representative, nor to be able to do the germination work to clear up those dormancy issues. But we did get to go to some pretty cool spots, which you can see in the photo on the right. So yeah, some of the limitations with threatened species collections is that it requires a judgment call on what is acceptable and appropriate for each individual species. So obviously when you're out collecting seed, you're removing that seed from the environment. Sometimes that's the appropriate call because in our case, where we're licensed to collect seed, we've been in consultation with everyone who's working on those plants, and we've made a long-term plan for conserving that species into the future. And we have a plan to do research that will help bolster those populations in the wild. It's the appropriate course of action. However, in some cases, it's better to not take the seed out of the environment. So some of the questions that you have to ask yourself is if there's evidence of recruitment, does the species actually produce seed, or will you need to get a livings collection team involved and actually get some of the horticulture expertise from the gardens involved? Has the genetics been done on the species, whether or not there's site accessibility issues, and just figure out if there's any difficulty coordinating research or collecting data and that kind of a thing. So in the last couple of years, we've actually made a bit of a switch internally within the Seedbank for our threatened species program. Because surely we would have collected all the threatened species by now if we've been working on it for 20 years, but unfortunately not. So we've changed tack to focus on fewer species, on making higher quality collections, and trying to actually tick a few of these species off instead. So the rationale behind that is that focusing on limited genera means that we can apply the learnings from the species within that genera more fluidly during a season, from collecting to lab work. So in this instance, we have had a focus on collecting grevilleas and homoranthus. And the idea behind that is that even though a lot of the threatened species are outliers for their genus, they usually do also have a reasonable amount of similarities based on the fact that, of course, they are taxonomically related and they do have some level of, similarity in structure and function. And so we use that to better target collections and seed processing and things like that. So it means that our work can be a little bit more efficient, and that hopefully has flow-on effects into improving the actual quality and efficiency of the collections. So the species often co-occur or occur in similar habitat and can be targeted in more efficient trips. And most of our target species this season are actually in the northern part of the state. So that means that it's a little bit easier for us to coordinate field work. So we have a multi-year focus this time. So as I mentioned previously, we had 60 species per financial year, whereas in this case, we have about 10 grevilleas and 10 homoranthus that we're focusing on, and we are focusing on them effectively until they're done. And so that means that we're focusing on them over multiple years, and we hope that that will yield a little bit more success in being able to actually tick these off and consider them as conserved. So we'll be focusing on fewer species so that we can ensure that we're getting representative collections completed before we move on to other species. And the germination workflows from last season on Grevillea species have already begun, and those findings will hopefully be more meaningful to interpret from a genus perspective, which means that because we're looking at how these individual species are responding, and we can then compare it at a genus level of, the other individual species that are threatened, we can kind of make broader generalizations on what specifically might be causing some of those issues in situ in terms of making those species vulnerable to being threatened. Yeah, so it's a bit of a change to our previous tack of just trying to get as much material as possible into the bank as soon as possible. But it's mostly just because we really want to ensure that when we have events like the Black Summer Fires, which happened in 2019, 2020, that some of these species that, we have small collections that we've made efforts to collect from, we actually have enough data and have enough material to be able to provide meaningful resources in their recovery. Whereas previously, because we'd sort of been stretching ourselves a little bit thin, we kind of had a little bit of everything, but we had a lot more to learn about most of those things. However, we still have time to tack on a few random individuals that happen to be doing well in a season. And one of those species that happened to be doing well at the beginning of this year was Aponogeton queenslandicus. And it basically is known as a holy grail species. Now, a holy grail species in the plant world basically just means that we've spent a lot of time looking for it previously and have failed to secure any material from it because it is just difficult to find, or the conditions that trigger it to fruit and flower are very restricted. So this is one of those species. It's an ephemeral wetland species. As you can see, there's some photos of it down in the bottom left. It's quite funky looking. It has a bit of an alien vibe. It's, neon green and yellow. And it pops up in one ephemeral wetland in New South Wales in west of Burke, which is nearly a 9-hour drive from Sydney. And it's actually the tip of the distribution of this species. So it does occur in the northern states and territories, so Queensland and the Northern Territory. But this is the furthest south it gets. So I've been out searching for it plenty of times before and never seen it. And we just so happened to get some excellent intel by James Barris of National Parks Northern Inland Branch in March of this year. And he had been doing some drone surveys and discovered flowering plants that occurred in those in that singular ephemeral swamp. And we were just super excited. He asked if we could come up and grab seeds. And of course we said yes, because we are plant nerds and we live for this. So we only had a small window of opportunity. And we basically did a crazy thing, and we did a 3-day round trip to Burke. So we worked out that it was around, I think, 25 hours of driving for about 5 hours on the ground of field work. But it was a really, really great time. And you can see my colleagues and myself to the far right top corner there. So Ruby, Steph and I hopped in a car. We drove up on a Monday to west of Burke. We met James out there. We got on our waders and we, tramped out to these swamps and had a poke around in the mud. So by the time we got up there, the plants weren't looking nearly as nice as they do in those photos in the bottom left. Those photos were taken by James. When we got there, they were looking quite dry and a bit sorry for themselves. However, they were producing fruit, which is very exciting because that's what we were there for. So we also managed to actually excavate a couple of the actual plants and bring them back to the Botanic Gardens of Sydney with us. And we have now those plants living in a little tank at the back of the nursery in Mount Annan. So I guess that's now their further south, southern extent. But that was a great effort. It was a really great collaborative effort, cross-departmental. And we now have a seed collection and a living collection of this species held at the Australian Botanic Gardens of Mount Annan. And I also included some x-rays of the seeds that we collected so that you could see sort of the variety of quality of seed, but also just the processes that we go through in terms of quality control. So on the left of the bottom x-ray, you can see that there's mostly good fill, which is because there's some nice bright white sort of coloring in the center of those little oblong shapes, whereas on the right hand side, you can see that they look a lot duller and they don't really have that bright white blob on the inside. So the ones that are on the left have good fill, they're viable seeds, and the ones that are on the right are mostly empty. So that's part of the process that we go through to sort the seeds before we actually store them in the seed bank, so that we know that the collections that people are taking out in the future actually contain viable seeds. Another collaborative project that we worked on as a team was securing some seed of the dwarf mountain pine for Fitzgeraldii. It was a multidisciplinary team, so it included RISA, which is the genetics team, the living collections team, which is the horticulture team, and the seed bank, as well as Saving Our Species, which is the threatened species departmental team, and national park staff and contractors. So it's a very difficult to access site. The ecosystems are very delicate, and there was a need to minimize disturbance. But we were able to use groundwork that had been provided by RISA because they had actually done the genetics prior, so they'd already gone through and tagged all of the plants for their genetic work, and that meant that we could just sample the seed from those individuals and keep track of them. So it means that we have really robust data for this species, which is really fantastic. So the species strikes easily and produces plenty of seed. However, none of those have actually eventuated into seedlings that have been observed in the wild. So we suspect that there's some kind of complex dormancy. So we went out, you can see Mira and Maz both standing underneath the spray of a waterfall making some collections. So they work both in the horticulture team. So we went out as a team together and I collected some seed and they collected some cuttings. And they have taken those cuttings back and already propped them up into little plants that are doing quite well and now producing their own seeds. Well, producing their own flowers. Not quite time for seed yet. The seed that we collected was the largest collection that's been made of this species so far. It was around 200 seeds. So we plan to revisit this species in the new year and have a bit more of a targeted approach of collecting. There's more than just threatened species out there. Although the threatened species are very important and of course threatened, they have, a high risk of extinction and they have a lot of urgency surrounding them. Because of that, they all occur in habitat of mostly common species. And unfortunately, most of the funding that we have received has been focused on the threatened species. And so we actually don't have a very comprehensive collection of the majority of our common species. So that's something that I'm very passionate about addressing as part of the work that we do, is focusing on improving our core collections of common species within the seed bank. And so that's another key change in our recent program, to focus more closely on collecting some of those species that are underrepresented in the seed bank. So you can see down the bottom a photo of a Beckia species, which is in the Myrtaceae family, located in Royal National Park, which is super, super close to the Botanic Gardens Mount Annan site. However, it just has never been a priority for us to collect, unfortunately, because of funding. And so, yeah, the first collection that we made of that was actually in 2021. You can see that it's some beautiful habitat, and actually in that photo to the right there, the majority of those species that are pictured in that photo are actually not held in the seed bank. So that's a little bit of a passion project of mine while I am the collections development coordinator, is to improve the core collections held at the seed bank, especially with, you know, individuals like this, the Beckia species in the Myrtaceae family. Some of you may have heard of myrtle rust, which has been impacting severely a lot of Myrtaceous species, and we just don't know the extent of that yet. We don't know if there will be other strains that will come to Australia at some point and impact some of these other Myrtaceous species. So it's really important that we do what we can to just ensure that we have the most representative collections of all of the germplasm available in New South Wales that we can get to. One of the things that we've also been doing a lot of this year is we've actually been doing seed conservation workshops. So seeds come in all shapes and sizes, and they take a range of approaches to successfully collect. We've been running workshops with increasing regularity to share some of the knowledge that we've developed on the ground. And we're more than happy to share what we've learned, and probably will learn something from you too, because generally the people who are interested in seeds, it's a fairly niche, fairly niche area. You guys tend to all have a huge amount of knowledge yourselves. So we prefer to run these workshops in our off season, which is late autumn into winter. And if that's something that you're interested in and you're based in New South Wales and part of like an organisation that works in the seed conservation space or ex situ conservation, something like that, then feel free to get in touch with me. Be happy to chat about that. And that is pretty much it. So yeah, we can just leave you with this lovely field of swamp everlasting to the left there. So that's actually a vulnerable species in New South Wales, and it was one of my favourite field memories to come across. It is, yeah, just a beautiful field of swamp everlasting that just spreads on and on in this random part of the south coast of New South Wales. So hopefully some of you will stumble across it one day. But I hope you have enjoyed hearing about all things seedy this evening, and I'm always more than happy to chat about seeds. So if you happen to be passing through New South Wales and passing along the M5, feel free to stop in at the Botanic Gardens and pop into Plant Bank. You can join a tour on Fridays and Saturdays that are hosted by volunteers. But if you see me and say that you have seen this workshop, I might even give you a tour myself. Cool. All right, I'll just finish on that. So thanks, Elle, for inviting us along to do this talk.
Thanks so much for being part of the program, Laura. We do have some questions. So one was, do you sell or gift seed to specific organisations or projects in New South Wales? I suppose that's kind of what we do with Seeding Victoria, we sell.
Yeah.
So, but part of my PhD, I did get some seed from the Royal Botanic Gardens in Melbourne. Yeah. So be interesting to hear what you guys do.
Yeah, it's probably similar to what you experienced, Elle, with the RBGV. So we do provide seed to researchers and external parties that are doing projects that are aligned with sort of the work that we're doing. And we have been exploring some avenues for that as well. Like I mentioned, we have been doing workshops. One of the recent workshops that we did was in the Central West with local land care groups in that region. That was mostly focused on actually empowering those local land care groups to actually make their own collections in a really robust way that meant that they could send in those collections to be held as duplicates by the Seedbank, and then we could release those seed out to them for the future, because obviously we have, you know, world-class facilities, and we can store seed under really excellent conditions for their longevity. So the short answer is sort of yes with caveats. So it kind of depends on the use of that seed. We kind of don't do it just for like recreation purposes, but certainly when it comes to like local land services, local land care, conservation focused things, and like university research and things like that, we can potentially release seed for those reasons. That kind of decision-making process is more so the Seedbank curator role, which is Graham Errington, who saw his little floating head in my presentation. He's really lovely. So, but certainly you can always send us an email and ask about it. And if we can't share our seed with you, then we can perhaps direct you to maybe someone who could, yeah,
Awesome.
Or advise on collecting that seed yourself.
Well, your seed testing information for specific seeds, is that documented for nurseries to be able to access? And I suppose now that you've popped your info in there, if people are interested and you do have that document, they can contact you?
Yeah, so you can contact me about that. Some of it is available online. The best place to look would be the Australian Germplasm Guidelines, which you can download a free copy, or you can buy it for around, I think it's like $35 plus, including postage Australia-wide. So that basically, a lot of the research scientists at the Botanic Gardens contributed to that tome. It's about 500 pages, and it has pretty much all the information that we could sort of think of to include about germplasm conservation. But yeah, in terms of those specific like dormancy breaking cues and things like that, we publish the reports that we basically get funded to produce for saving our species and things like that. You can sort of keep your eye out for that, but it should also be accessible on our website. Some of that should be coming out over the next year or so. Some of it has been a little bit undocumented or just sort of filed away, and so we've been having a bit of a push to make it more publicly available. So you should see more of it coming available, but if you're struggling to find any information, then just also feel free to reach out and we'll see if we can help out.
Awesome, thank you. The next question was also one that I had. Wondering what you mean by maternal lines of collections and what makes maternal lines more robust.
Yeah, cool. Okay, so maternal lines basically just means keeping collections from individual plants separate. So rather than making a collection from a population and pooling all of the seed that you make from, 10 or 15 or 60 individuals into one collection, you actually keep the seed from each mother plant or maternal line. The reason that we do that mostly for threatened species is related to genetics. So it means that if we want to down the line, we can actually test that seed or the germinants of those seeds for genetics. And the reason that we consider it to be more robust is just because it gives you a lot more flexibility in terms of what you can then do with the seed down the line. There's a lot of variety in what you can do in the seed sort of production space and things like that, seed conservation, and so you really have to think about what your actual outcome is, what the purpose of collecting that seed is, and then distributing it. For us, we basically want to have these seed collections made, but we don't necessarily always know what the future use will be, and so I guess we consider the most robust kind of collections to be the most flexible to any future use that could come up. So yeah, it's basically, but just keeping those maternal lines separate. If you talk to any of our genetics team, they will say that is in fact the only way that we should be making collections. But for those of you who have actually been out on the ground making collections, you will understand that there's a lot of logistic difficulties with that. And also that in terms of actually doing our germination workflows and testing the seed, we actually do sometimes need to pull that seed so that we have enough to actually infer meaningful data out of, like, to get meaningful data out of that, because otherwise we just don't have like the replication that we need. Yeah.
That's it. Okay, well, thank you. There's a couple more questions. What type of agar is used to germinate seeds, and does agar need to be tailored to get optimal germination?
So I mean, you can buy like agar to eat. We just buy. I'm not honestly super sure where we source it from, but we just basically get lab grade agar. And I think that it's effectively the same. It's just not food safe. So it's just less processed. And
So don't
having
eat
gone
that
through
one,
that, yeah, don't eat that one. But it basically is just, it's just normal agar.
We also had a question about taking cuttings of threatened plants, and
Yeah.
I did see that you had done that with the dwarf pine. Is that also common, or is it sometimes a better way, or is that preferred sometimes with certain species, or not preferred?
Yeah, absolutely. So I mentioned at the top of the presentation that seed is kind of our preferred method for conserving species, just because it's the most cost effective and space efficient way of conserving species. But there are a lot of species that it's very difficult to either collect seed or germinate seed. In that case, we might go to cuttings as a sort of backup. I did not include this as an example, but perhaps I should have, because it would have answered this question quite nicely. But there was a species, Melichris gibberigi, that was a critically endangered species, I believe, that we'd tried to make collections of in situ multiple years in a row. We tried to collect seeds, we hadn't been able to. It's an apacrid, which are notorious for being quite low in producing seed, and also having complex dormancy. So what we actually did end up doing was collaborating with the living collections team at work, and we did get some cuttings and take them back to the gardens, and they grew them on in the nursery, and we basically had a mini seed orchard area for that species. And within a couple of years, we were actually able to collect several thousand seeds from that species, which was more than we'd ever been able to achieve from the wild populations, because they were getting TLC that obviously they would never get in the wild. And so that meant that we could then go on and actually test the seed. So yeah, there are definitely circumstances where it's better to take cuttings. There are species that we can only take cuttings of. A lot of rainforest species, you can't conserve the seed at all because they have too high a fat content, basically, and they won't tolerate drying or freezing, which are both essential for orthodox
seed storage.
Seed storage, yeah.
That actually segues really nicely into our last question, which was my question.
Yeah.
Would you mind just giving like a brief definition on orthodox versus non-orthodox seed?
Yeah, absolutely.
For listeners?
Yeah. So orthodox seed is something that we consider able to tolerate drying down to a low moisture percentage, typically between sort of 3 and 5% moisture content, and also tolerating freezing down to minus 20 degrees, which is how we store the majority of, sort of up to 80% of Australian species are stored in orthodox conditions, which is in a minus 20 degree freezer. Unorthodox species are basically all of the species that don't fall into that category of being able to be dried down or frozen. Some we have recently found will tolerate freezing up to minus 5 degrees, but not below that. And so we're actually learning the sort of limitations for some of those species, and hopefully we'll be able to cater to that a little bit into the future. But yeah, it basically just means can tolerate those standard drying and storage conditions, or cannot. And the things that cannot are typically like orchids, rainforest species, things with really big, fleshy fruit or seeds.
So thank you. And I think I just wanted to mention one more thing before you leave. You did mention, like, how haven't you collected all the threatened species by now? But just wanted to highlight as well just how important that work is, as well as your new work in the species that aren't threatened yet, because, you know, species are always they're not static. And especially with all the issues that we have these days with the environment, things are becoming more threatened as much as we'd like them not to be. One more question. We're being told to bring species down from Dubbo for future climate plots, but I've had difficulty sourcing seed from that area. Is this the reason you would sell seed for?
Not necessarily a reason that we would be able to provide seed. We don't really sell seed because we're, you know, a New South Wales department organisation. It's kind of, you know, public service. We don't really do that. And we also don't necessarily have collections for the species maybe that you're interested in from Dubbo. Like I said, we do lack some of the common species from a lot of New South Wales, unfortunately. But it would be a great reason to get in touch with, I think it must have been the Grenfell local native nursery, which is one of the sites that we visited on our recent workshop, sort of Seedbank roadshow that we did. And they may be able to assist you because we were talking about assisting them in improving their seed collection strategies for a lot of the species in their areas and trying to support them improving their collections. And that would be from around that sort of central west area that would include Dubbo. So I think that probably the main way that we could assist with this kind of a thing, just like I mentioned, we don't tend to make huge collections because we focus on quality over quantity, is that we can probably assist in maybe linking people up, but certainly we have been having a lot of those discussions about where to source seed from when you're being told to collect outside of your area for climate.
Thank you so much, Laura. And just for anyone who is listening and not reading, Laura's email is laura.watts with 2 T's at botanicgardens.nsw.gov.au. Otherwise, feel free to shoot me an email at info@seedbank.com.au and I can link people up or answer more questions. Thank you, everyone, for joining us tonight. And thank you, Laura, for taking time out to join us. And hope to see you all at the next Seeds of Knowledge webinar.